Okay, so welcome everybody. If you can mute yourself and then unmute if you have questions. Don't be afraid to interrupt me. This is our third lecture. Last lecture we talked about immune privilege, blood-brain barrier, and what immune privilege is. And today we're going to talk more about cell types. So today we'll focus mostly on cell types of the brain. And I'm not going to re-explain to you what a neuron does and what an astrocyte does. What I want to do is tell you about these cells in the context of why they're relevant to neuroimmunology. And I have to tell you some basic function of the cell so that you can understand it. And I want to make clear that there really is only one immune cell type in the brain, right? And that's the microglia. Okay. Somebody's unmuted and it's bothering me. All right, I just muted everybody. I don't know if that means you can unmute yourself. Maybe not.
We can.
Okay. Okay, so here's what we want to talk about today. The cell types involved in neuroimmune interactions. So within the brain, we have the neurons, which are involved in brain to body, body to brain, neuronal connection, oligodendrocytes, astrocytes, endothelial cells, and then the ones really highlighted here, the microglia and CNS macrophages. And the reason I'm highlighting them is that really they're the main immune cells, right? People will talk about immune functions of these other cell types, and not to lie to you that there is some, but really the specialized tissue macrophages are the microglia. And then next week we'll talk about these peripheral immune cells that oftentimes interact during damage or disease or trauma, or sometimes in just normal healthy immune brain interactions, neutrophils, lymphocytes, and mast cells. We'll talk about those more. And we may not finish today. Like I said, I only want to go, for 1 hour, because I think that's longer than that is too long. So we might finish kind of in the middle of the microglia section. I put the microglia last just for that reason. So I do want you to sort of understand this slide. And this goes to a lot of understanding, why microglia are different than these other cell types. Unfortunately, let's see, my face is in the way of actually seeing the slide. So let's go over here on the right first on the CNS cells. So neurons, oligodendrocytes, and astrocytes all come from the neuroectoderm or neuroepithelial progenitor cells. Let's see, I'll put the little highlighter on. Where is it? Okay. So right here, the neuroprogenitor cells, and they give rise to neurons, and then macroglia, which are the oligodendrocytes and the astrocytes. So what this slide is showing you is that here in the hemopoietic cells, so most of you, or hopefully you know that all of your immune cells, your peripheral immune cells, and your red blood cells come from the bone marrow. And these are the hemopoietic stem cells. Hemopoietic is just, blood origin. And there's common myeloid progenitors and there's common lymphoid progenitors. So let's start here with the lymphoid progenitor. They give rise to lymphocytes, which are the T and B cells. The common myeloid progenitors here, early in development from the embryonic yolk sac, they give rise to these long-lived tissue macrophages, not only in the brain, but in several tissues, including the liver and lung. And so microglia actually come from this embryonic yolk sac, and they come to the brain early in development. In rodents or in mice, it's embryonic day 8. I think rats, it's somewhere in that same range. And they've even seen evidence of this happening in humans. And so the microglia come into the brain. They're from this common myeloid progenitor. And because they develop in the brain, they have this specialized profile, which we call microglia. Now, what I think is oftentimes confusing for people is that, when we also look at what happens to a common myeloid progenitor, you get this, they become these granulocyte myeloid progenitors and myeloid progenitors. So the granulocyte myeloid progenitor becomes granulocytes, which are the cells that can degranulate, like mast cells and neutrophils. And then the myeloid progenitors become dendritic cells and macrophages and monocytes. So within the brain, there's also an area where we see these CNS macrophages, and they're not in the parenchyma. So remember, I asked you guys to remember what parenchyma meant. So they're not in the tissue, they're not in the meat of the brain. They're sort of in specialized areas. And you'll find these monocyte macrophages in the meninges, like we talked about Tuesday, the meninges, the choroid plexus, and the perivascular space. So keep that in mind, that these two cells, the microglia, are the permanent residents within the brain, and these CNS macrophages are also contribute to some of this macrophage pool within the brain. They're in lower amounts, they are in specialized areas, and these two cell types are cousins. So they have a lot of activities that are related, but some are specialized, and the ones that are more specialized would be the microglia that have come into the brain during development. So I want you to consider this slide and think about what this means. So let's talk about these cell types first, just because I think they're a little bit more straightforward. None of them are immune cells. They all come from these neuroectoderm progenitors and represent, what helps make the brain and spinal cord function appropriately. Let's see, I think I already talked about these things. So let's start with neurons. My little stick drawings of neurons, are actually a pyramidal neurons within the hippocampus. That's sort of the drawing that I can make. They're right here. So my little stick drawings are right here. These are sort of the ones you'll see in the hippocampus. But there's a large diversity of neurons within the CNS. Many of you probably know that there's excitatory, inhibitory, and interneurons. We see, the Purkinje cells, which are specialized in the cerebellum. We have cells that are specialized in the thalamus, cells that are involved in motor axon movement. So we see a lot of diversity, and these are all just obtained from Golgi stain and actual tracing. So in terms of what a neuron does in neuroimmunology, it really doesn't have a role in that it's not an immune cell, but it is involved in neurotransmission, right? So from body to brain, brain to body. And in this function, we see that one of the things that neurons don't like, let's see, I have this stupid thing in front of me. Okay. Neurons do have the capacity for what's called the expression of MHC class 1. Now, MHC class 1 is something that all nucleated cells have. It's how our cells declare self. So they present self-antigen on this MHC class 1. This is something that we'll talk about when we get a little bit more into the immunology parts. But every nucleated cell will have this, and it's a way you present, so the immune cell knows that you're part of your own body. So neurons have this, and it's more inducible. I don't think you always find expression on it, but they do have it. And this means that if you have a CNS virus, that neurons will detect or will show the immune system that there's something going on. Neurons also like to stay in that homeostatic, interstitial fluid CSF environment that has, the optimal buffer for neurotransmission. It really doesn't want to see T cells in the brain parenchyma. And so it turns out that neurons express Fas ligand. And what Fas ligand does is it causes immediate cell death into T cells. So neurons create an environment that's unfavorable for things like T cells going into the brain parenchyma. We also see neurons are very important in the regulation of microglia. We're going to go through this quite a bit in some of the later lectures, so we'll just touch on it here, that we see a number of things that are involved in contact inhibition. The CD here means clusters of differentiation. And if you see CD in front of a number, it's usually sort of tied to the immune system. So they use the CD200 on the neurons, and then the receptor is on the microglia, and it helps with contact inhibition. It turns out that neurons do a lot to keep microglia in check. The other thing that neurons produce a lot of is this thing called fractalkine, which is the CX3CL1, which you see kind of in that chemokine state, very highly expressed in the brain, and it seems to, again, inhibit or shut off glial responses under normal conditions. There is evidence that neurons will, when they're damaged or infected, that they're going to secrete factors that influence other cells around them. So it's like a warning response. They can things like interferon gamma, substance P. When neurons also dump things indirectly into the cytosol, like if they've been damaged by an injury, there'll be higher levels of glutamate, laminin, alpha-synuclein, and these things can activate microglia and astrocytes and recruit other immune cells. And probably the most important thing that we want to talk about in this class, in terms of why we talk about neurons in neuroimmunology, is that neurons have cytokine receptors. Remember from the very first lecture, I told you that cells, the reason that neuroimmunology works, that immune cells and brain cells can communicate, is that cells of the CNS have receptors for things from the immune system. So that neurons have cytokine receptors. And those cytokines are functional, meaning they change the way the neurons fire, they change the way they develop, they change the plasticity. So if you ultimately want to affect behavior with this inflammatory event, you ultimately have to work on the neurons. And cytokines have that ability to change the way the neurons fire directly. That's really a key component I want you guys to think about. So that's my kind of overview of neurons. We'll definitely talk a lot more about them because they end up being the end target for a lot of things, right? So neurodegeneration, for example, what's happening, right? It's death of the neuron. So ultimately, they are the sort of end of the road for some of these neuroimmune responses, especially in disease. So next up, we want to kind of start going through these oligodendrocytes. And oligodendrocytes are important for myelination. And does anyone know what the counterpart of an oligodendrocyte is in the peripheral nervous system?
Schwann cells.
Schwann cells, right? Now, is there any difference between the number of axons that a Schwann cell wraps versus the oligodendrocyte?
Oligodendrocytes can wrap multiple axons,
And Schwann cells can wrap
just one part of a neuron.
Very good. Yeah, so that's absolutely right. So take note of that. Those of you who are non-neuroscientists, so oligodendrocytes are specialized. And here's a picture of one. So you see one oligodendrocyte here wrapping multiple axons. So these linear tracks here are the axons. Oligodendrocytes constitute about 15 to 20% of all glia. They're found in the gray matter and the white matter. Do you guys know the difference between white matter and gray matter? Yes? Okay. So white matter is where all the sort of myelinated tracks are. So these guys come from the neuroprogenitor. The resident is within CNS, and you're going to see a lot of different ways people detect them. So you can detect them using certain enzymes. Luxol fast blue is one of them that people look at myelin. People also look for proteins associated with myelination, myelin basic protein, protein lipidase, and then they'll look for things like progenitors, so NG2. And when we start doing some of the applied, you might see more of these markers. In terms of what oligodendrocytes do in an immune function, is that, well, they're important for myelination, but this myelination event can be affected by cytokines and chemokines and growth factors. So If you guys know of multiple sclerosis, you know that you have a demyelination. So the cell that ultimately is affected is the oligodendrocyte. So they take a lot of cues from astrocytes. We see that a lot of times in inflammatory conditions, you find these cells co-localized with active microglia. The other thing that happens is that oligodendrocytes are important for repair. So if you have damage to the central nervous system, you can see that oligodendrocytes, part of what some of these cells are doing in terms of neural repair is trying to get oligodendrocytes to proliferate and then remyelinate. So you see a lot of diverse and interesting signaling between microglia and oligodendrocytes, or astrocytes and oligodendrocytes, in terms of repair functions. And we know that they produce some heat shock proteins. My guess is this is part of that damage response, letting know that other things are going wrong. And then what we see a lot in the context of neuroimmunology is that oligodendrocytes are very sensitive. And they're sensitive to really three things, I think, that are relevant to us in our lectures. They're very sensitive to high levels of glutamate, nitric oxide, and to TNF alpha, which is tumor necrosis factor alpha, which is a inflammatory cytokine. So in a lot of end results of disease or infection, we see loss of myelin, and you can even see loss of myelin with a traumatic brain injury as well. So this is a little better picture of an oligodendrocyte, much better than the one that I drew, but you can see the cell body and then all its little processes that are wrapping axons. This is what an axon looks. This is what's called ultrastructure. So you see these thick bands of myelin. Okay. And so we should talk about this a little bit, because this becomes important, in context of what inflammation and things are doing to neurons and oligodendrocytes. Is why is myelin important? And if you probably haven't taken any neuroscience classes, maybe it's not as obvious. But the reason we have myelin, and I think you can see here, is that it wraps, and it wraps parts of the axon, right? So it doesn't wrap all of it. And what this does is forces for this cell to send a signal down the axon. The signal kind of jumps through these nodes of Ranvier, and this action potential moves down. You get this what's called depolarization. You need channels to do this, but the signal jumps down. And the reason that we have this is speed. So this saltatory conduction is this fast jumping conduction of the nerve signal. So it leaps from node to node. It's very fast, and you see these channels are needed for this depolarization. And so you see an unmyelinated axon still can do an action potential, but moves slower. Now, I have another question for you guys for some extra credit. Are all of the CNS axons myelinated?
No.
That's right. So not everything is myelinated. So you do have a bunch of unmyelinated fibers within the brain. So let's see. The ones that are myelinated are going to move faster. So you're talking a lot about the motor neurons, right? So all your motor neurons for movement are going to be myelinated. These are just showing you some of the pumps. So these are sodium potassium pumps. They use these gradients to create energy in the form of ATP. And this is what allows you to have this depolarization. And you find these channels sort of in those in the nodes of Ranvier. So you see them kind of jumping through there. So this myelination, not only does it increase the speed of conduction, it also makes the cells signaling more energy efficient. And that's important because I've told you that the brain uses 20% of our body's energy. And so you need ATP to move sodium back into the extracellular space and potassium back inside the axon. When you have myelination, therefore, so there's less sodium needed for action potential. So it's, again, Dr. McTighe, when she provides us lectures on this, which I don't think she's doing this year, but normally she does, she'll talk about this being green energy. This is also important, I think, in the context of oligodendrocytes. There is quite a bit of turnover So you have over the lifespan or just over normal health, you see that there's progenitor cells. And so you can label these by different antibodies, and I don't think it's really super important for us to know, but you might see some of these different labelings depending on what kind of papers we do for the discussion periods. And oftentimes, or all the times, I let the instructor pick those papers, so I don't know them often ahead of time. But we may see some of these things where we see, we're looking for the progenitors, and then as they develop, the oligodendrocytes change quite a bit. So you can see that on the surface. They become more mature. And it's the mature oligodendrocytes that you see the myelin basic protein and PLP on there. Earlier, they have things like the platelet-derived growth factor receptor, nerve growth factor receptor, and so they change over time. And so I think that the point I really want to make here is that this process is both negatively and positively influenced by inflammatory mediators. So a lot of times when you're trying to enhance repair, there's mediators that will increase this process to try to remyelinate under damaged conditions. And then a lot of times under sort of really high inflammatory conditions, this process is halted. So not only you might cause demyelination and death of oligodendrocytes, but you also may inhibit the progenitors from differentiating. This is the same idea, but using my little stick drawing, going from progenitors to the pre-myelin oligos, which would be Ng2 negative, oligo 2 positive, and myelin basic protein negative. And then sort of as they start to myelinate, you see my cell here myelinating multiple cells. And you see that now they're the mature oligodendrocyte. That's sort of the one I showed you a picture of. And they're going to be Olig2, Ng positive, Ng2 negative, GFP negative, and myelin basic protein positive. Again, I'm not going to ask you all of these things. I'm just putting them there so you can see, and that you might see this again. And again, things can positively and negatively influence this. One thing that's been pretty clear in the oligodendrocyte field is that TNF alpha, which is one of the kind of inflammatory cytokines, negatively impacts this myelination process. The other interesting thing is iron is a cofactor for this. And I know in some of Dr. McTighe's work and others that iron is if it's not tightly controlled or sequestered, iron is very negative within the CNS. The other interesting part about brain development and early life responses in terms of infection is that microglia are involved in sequestering this iron and giving the iron back to the oligos. And this process often goes awry in infectious conditions, which might affect brain development. And someone like Stacy Bilbo might talk a little bit more about this process. Okay, so things I think are important to know about oligodendrocytes is that oftentimes they are damaged or die with inflammation, very sensitive to excitotoxicity caused by glutamate. You see a lot of them die by oxidative damage. So if you think about inflammation, not only there's cytokines, but there's also oxidative stress. We know things like pro-inflammatory cytokines affect the myelination process, but also can directly kill oligodendrocytes, things again like TNF alpha. You also see indirect killing by glutamate toxicity and indirect killing by other sort of pathways. And what's key here is that when you start to lose oligodendrocytes, you can have axon swelling, loss of nutrients delivered to the axon, and then microglia activation and axon degeneration. So once you start losing axons, there's really no replacement for those, right? So that becomes a big issue. This is just showing you the idea of excess glutamate, which I think is has a pretty major role in inflammation and sort of this idea of secondary damage. I think I talked about this, at least hinted at it, this collateral damage that inflammation causes in the brain or spinal cord versus collateral damage that inflammation has on the skin, right? So here we're talking about excess glutamate can increase intracellular calcium. You have this whole processing happening within an oligodendrocyte or other cells for that matter, where calcium is going to increase arachidonic acid production. We get you more free radicals. You have this nitric oxide synthase. You can have things that are like proteases increased and mitochondrial damage. And so this glutamate pathway feeds into excess calcium, and then we have more free radical damage, DNA damage, and mitochondrial damage. So this is sort of a a very negative signaling pathway for oligodendrocytes. Everybody probably, I didn't mention this, and I'm sorry, but glutamate is one of the really abundant neurotransmitters. It's tightly controlled. We'll see in a few slides here that it's tightly controlled by astrocytes. But you never want too much of it around because it's excitatory. And so you don't want it sitting. And so the release of it during damage or infection can cause a lot of problems. And part of that is sort of kind of tied to the mitochondria. And mitochondria has too much calcium that enters the cell. This is when you can start getting those apoptotic pathways. What you get this is sort of describing apoptosis. You guys have probably heard about this in some of your classes already. Apoptosis is this, comes from the Greek word from falling leaves, I think, but it's this programmed cell death pathway that involves mitochondria producing factors that activate these specialized enzymes called caspases, and it leads to this cell death protocol, this nice programmed cell death where the DNA ends up being chopped up in 200 base pair fragments. And just out of fun here, anybody know why the DNA shows up in this nice 200 base pair ladder fragment? If you were to run it out on a DNA gel?
Probably because of nucleosomes. I mean the chromatin.
Yeah, exactly right. Yeah, it cuts in between the nucleosomes, so that you have this. What wraps around a nucleosome is about 200 base pairs. So it's pretty defined sequence. So you can oftentimes tell if a cell is apoptotic if you were to homogenize it, get the DNA, and then run it on a gel. You'd see these really nice, tight 200 band fragments. Okay, so oligodendrocytes, the key factor here, without getting lost in the weeds of what I went off on that tangent, is that they're sensitive to this excess glutamate, can cause programmed cell death that's going to involve the mitochondrial responses. And we see this, we see glutamate. You can add glutamate directly to cultures of oligodendrocytes that have been differentiated. And you see damage to myelin, you see increased glutamate, you see what's called spectrin breakdown, which is involved in structure. So showing control versus glutamate. And so we see this pathology in a lot of different conditions, including multiple sclerosis, where you see inflammation, immune cells, oxidative stress, and excess glutamate. Let's see, this is also kind of tied to glutamate, and the idea glutamate also can activate some of the immune pathways as well. So it can be a fact. Glutamate is something that other cells will sense. If it's released from dying neurons, it will activate microglia and macrophages. This is going to increase inflammation. Things like TNF alpha, which you've talked about, which has been studied pretty heavily with the oligodendrocytes. We know that there's receptors for TNF alpha, and it can affect the oligodendrocytes negatively. Okay, so I think that's what I wanted to kind of cover with oligodendrocytes. And again, this is not going to be the first time we ever see these cells. We're going to see all of these cells over and over again. But this is just a little introduction to get your feet wet and to start telling you some basic things that I want you to know and will help you understand. Let's see. So the next one are the endothelial cells, and these are the specialized endothelial cells. They are part of the blood-brain barrier. They're going to have these tight junctions, and they're key in the structural barrier of the blood-brain barrier. They're also going to be involved in nutrient transport. So one of the things I've told you before is that the brain uses a lot of glucose and has glucose transporters that are non-insulin sensitive. Does anyone know why that's important? A non-insulin sensitive glucose transporter?
Is it because your brain always... Oh, I hear my echo on Courtney's computer. Is it because your brain always needs fuel, aka glucose?
Yeah, absolutely. Yeah. So other tissues will be insulin sensitive, meaning that your body will know and want your body to pick up glucose when you have excess glucose. And so there'd be inducible, insulin sensitive glucose uptake, but the brain is always able to uptake glucose independent of insulin. And you can think about that as, think about people who have type 1 or type 2 diabetes, that, the brain is not starving of glucose. It's all the other tissues. Or have too high glucose, for that matter. Okay, so in terms of immune function, all nucleated cells, again, will have this MHC class 1 expression. This is a major histocompatibility complex. We know that they're also going to be important in preventing soluble immune factors cannot cross the blood-brain barrier, and cytokines do not cross. It's going to force things when things do cross. They generally cross in between here. They kind of come between these tight junctions. There's also evidence of a lot of cytokine receptors on endothelial cells. So things like IL-1, TNF-alpha, and then also people believe that there's cytokine transporters that transport cytokines across. You're also going to find these things called integrins and selectins, which are adhesion molecules. So if you end up wanting to get cells into the brain, you have to have a way to stop them in the blood. Think about your blood flowing through your body. How does a cell stop at a specific site? It has to adhere. So adhesion molecules are important. And we have a whole lecture on trafficking and adhesion, so we'll go through that. This is just kind of showing you the different ways that the endothelial cells are happening here. This transcellular traffic, so this is the flow of the blood. The red here are the red blood cells. Now, if you have high levels of cytokines in your bloodstream, for some reason, you're going to have cytokine receptors within the endothelial cells here that transmit signals across. We'll talk about in pretty good detail how this happens. These number 2 is a proposed cytokine transporter that would transport a cytokine like IL-1 across. And then this number 3 is the selectin expression. So this slows the speed of white blood cells through the blood, and eventually you get this tight adherence. This would be the step you needed prior to a cell kind of getting into the brain. And when they get in the brain, they act like I said before, they usually get stuck in this perivascular or Vacno-Robin space. And this white bar here again is that glial limitin. And just asking a question, if you guys learned anything from last lecture, who makes that glial limitin?
Astrocytes.
All right. So yeah, absolutely. Good job. Okay, speaking of astrocytes, so again, we're going to see a lot more of endothelial cells. Astrocytes, I would say we're starting to get a little bit closer to some immune function. Astrocytes have some capacity, but again, they're not immune cells. They're derived from the same progenitors that neurons are. And so normal functions, physical support, they're important for the blood-brain barrier. They're very important in neurotransmitter metabolism, which we'll talk about. They're very important for what's called neurotrophic support. For those of you who are non-neuroscientists, what neurotrophic means is growth factor. It just happens to be neuron growth factor. We also know that they're very important in this kind of neuronal synaptic transmission. Astrocytes really constitute the majority of glia, and astrocytes outnumber neurons 1.4 to 1 in humans. So there's actually more astrocytes than neurons. So there's a little bit different constitution when you're talking about rodent brain versus humans. Sometimes something you need to consider, especially as we do a lot of our discussion papers. In rodents, 30% of the CNS cells are astrocytes, and humans, 50% are. So there's more glia. It seems like the higher order you move up in mammals, the more astrocytes you have. So they come from a neuroectodermal lineage. Where do you find them? You find them in both the gray and white matter. How do you detect them? So in some of the papers that you'll read and things that you'll look at, people will use glial fibrillary associated protein or GFAP. So you'll see that one a lot. ALDH1L1 is another very popular one. Anyone ever stained for GFAP in a rodent brain?
Not in rain, but in a retina.
Have you, as anybody who's looked for it, found that GFAP is not expressed everywhere in the brain?
Yeah, it's definitely not the entire astrocyte.
Right. So it is a little bit of a factor when people use only that one, because it only labels, it seems to be region dependent, particularly in rodents. So like you'll see almost no cortical GFAP expression, but you'll see it in the hippocampus. So things like these ALHD1L1, or what are called, they're glutamate receptors, they're EAAT2, and there's a bunch of different ones. They're more reliable because they're on all astrocytes. They're just a little bit harder to label for. And then we see things that are also kind of region dependent, vimentin, aquaporin 4. Aquaporin 4 is a very important water channel. It becomes important for some of the glial lymphatic stuff. Vimentin is a structural protein. Oftentimes see it increase with inflammation or scarring. And there's a numbers of different astrocytes. And so I want to kind of discuss these for a few minutes, because it gets into a little bit more astrocyte diversity. And I think astrocytes in neuroimmunology are just really starting to be looked at. And I think it's because if you look at what's happening with the microglia in any condition, and I know many of you study microglia, but if you don't put astrocytes in that equation, then you're really missing something. All right, because there seems to be a lot of connection between astrocytes. So it's important for us to kind of think about astrocytes and also talk about the little bit of diversity what they have. So these astrocytes here, so the first one is these protoplasmic. And so the way I've written, now these are all hand drawings by me, so you can yell at me if you'd like to, but this is showing you that. And so if you were to label astrocytes in the hippocampus, you would just see a star. So astro means star. So you see these really star-shaped, but they're really not star-shaped. They actually have a protoplasm around them, and they have a defined space, but you don't see that with the different labelings. You find most of these protoplasmic astrocytes within the gray matter. It's the most prevalent astrocyte. They have morphology associated with thick, short primary branches, originating from the soma, the divide into secondary branches and leaflets. These are the ones that you probably find encasing the blood vessels around the blood-brain barrier, also around synapses, and important in the glial lymphatic drainage. And these are the ones that are basically going to be ALDH1L1 positive and GFAP negative. Then you have these fibrous ones, okay? And these are going to be found mostly within the white matter. And you're going to find that they have close contact with the nodes of Ranvier and blood vessels. They have longer, thinner, less terminal branching. Notably, they're found in CNS tissue, and you'll find these guys, the fibrous ones, in sort of the astrocytic scar. You're going to see these are the ones, the fibrous ones are going to be the ones that are GFAP positive. So these are the ones you see after injury and things that are very fibrous. And then you have these radial. They're apparent in development. They're important in the scaffold system to help neuronal migration, and also involved in it can transform into what's called a stellate astrocyte.
Does the protoplasm in the protoplasmic astrocytes have a particular function that it's useful for those? I mean, is there a reason that it has that distinct morphology from the other ones?
Well, I think it's because astrocytes do not normally overlap in any space. So it seems to be involved in the correct placement. So it's to prevent sort of overlap, I think. So you'll see overlap of these fibrous astrocytes, but you won't see overlap of the protoplasmic ones. And I think there's probably a reason for that.
Okay.
And then Isabella can correct me if I'm wrong, but the radial astrocytes are the ones that are specialized you see in the retina that are called Microglia, and then the Burfman glia in the cerebellum.
Yep.
Good. Okay. Okay. So astrocytes and neuroimmune events. And so now we're getting into a cell that has a little more neuroimmune function aside from just being kind of the end result or being involved in subsignaling. We see that they are involved in the blood-brain barrier. We know that they support growth factors for neuronal support. So neurotrophic factors or growth factors, they're involved in synaptogenesis and neurogenesis. That synaptogenesis is the formation of synapses. Neurogenesis is the formation of new neurons. Another really important thing is they act as a glutamate sink. So I just went through some slides this morning talking about glutamate and glutamate excitotoxicity. Anyone know what astrocytes turn glutamate into?
So I'm guessing from my biochem, is it alpha ketoglutarate that they turn it into?
Glutamine.
Glutamine. Okay. Well,
So it's non-reactive. So basically, they're basically detoxifying it. So you think about this, if you start to disrupt this astrocyte biology, you're also in turn going to increase glutamate, which is going to can affect your oligodendrocytes, right? So sometimes when we think about collapse of a CNS system with trauma or infection, it starts here with some of the problems with the astrocytes. Okay, so in terms of neuroimmune events, we know that they have this really tight interaction with microglia. Fancy word for that is called modulation. We know that ATP activates the calcium wave, and we see that this calcium wave really sort of spreads across astrocyte activation. There is some evidence they produce inflammatory cytokines, and I have this in here. You can see evidence of them making some cytokines. The other thing we see is proliferation, which is also called astrogliosis. This is when we see that sort of expansion of this fibrous looking astrocyte. There used to be this idea that astrocytes could present antigen on something called MHC class 2, which is the one that's involved in more of that adaptive immune response. And we'll go through this, but they can't. They don't have all the machinery to do this. They can also initiate clotting cascades. So you want to stop bleeding. Astrocytes are important for this. They also do something called scavenging of free radicals. So again, like glutamate, you don't want to have all these reactive free radicals floating around, and they can uptake those and detoxify them. And we also see astrocytes important for glial scarring, which is walling off areas of damage. For any of you guys that study head trauma, spinal cord injury, stroke, or CNS infection, you're definitely going to see evidence of glial scarring. And sometimes even humans will go show up and have a scan, and someone will say, "Do you ever have you don't even know they had a bacterial infection or something?" They'll have like this area within the brain of this little capsid of reactive astrocytes. I have no idea. So you see scarring quite a bit in different situations. We know that they can be activated so astrocytes definitely respond to cytokines. They respond to damage-associated proteins. They respond to ATP through what are called purinergic receptors. They're very responsive to calcium, glutamate, and a number of chemokines. Okay. So a little bit more of this idea is that them being involved in complement cascades, cytokine production, chemokines, secondary immune molecules like prostaglandins, nitric oxide. And so this is a personal disclaimer because I've done a lot of work on astrocytes and we've done a lot of single cell sequencing. And I really don't think astrocytes make many of these cytokines and chemokines. They respond to them for sure, but I don't think they make them. And the reason is that they just don't express them. When you actually look at them and you don't have any confound of macrophages or microglia in there, and they certainly help microglia make cytokines, but they don't seem to make them. And that's just my personal opinion, but I will not let that interfere with the data that's out there, meaning that you definitely find papers that astrocytes make all these things, and maybe you've seen it in your own lab. I just don't think they really do, but I won't let that interfere with what I'm teaching you. But don't pick a paper about astrocytes making cytokines for, no, I'm just kidding. You can pick whatever you want. So anyway, so here's astrocytes. You've seen this picture before. You've seen astrocytes here as part of the glial limitin. If you didn't know that, some of your student friends just told you that. And here's an actual picture. This is an actual labeling here. This red tube, it kind of looks like a lip here. You see these little cells in red, and you see the black here is that astrocytic end foot, all lining that blood vessel, or actually be lining where the endothelial cells are. This is showing you what a glial scar looks like. In the white matter, you see, this really dense fibrous layer of these astrocytes. And the thing about these glial scars, and a thing that people are very interested in, is that it's a mechanical and chemical barrier. So this astrocyte, in the context of injury or infection, will proliferate. And so now you'll see this kind of dense overlap, which you normally don't see of astrocytes, this dense overlap. And when you're thinking about neuronal repair and axon regeneration, this is a mechanical barrier to the neural growth cone. All right, so as the scarring happens, you see this mechanical barrier, and also becomes a chemical barrier. So moving axon growth cones through a glial scar is a really important area, and people are interested in that in the context of spinal cord injury, as you can imagine. And it's another really good topic for you guys looking for topics. This is some papers showing, this is done by Soforno's lab at UCLA. He's come to talk to us at OSU before, a really great astrocyte biologist, and has some really cool tools, been looking at astrocytes for a long, long time. This is one of his kind of landmark papers a long time ago now, but he had this cool model of this astrocyte on a death domain. So basically was able to kill astrocytes. This is a GFAP, this herpes simplex virus TK design. Basically, if you give the mice ganciclovir, TK, thymidine kinase, will create a toxic metabolite and it'll kill astrocytes. This is one of the first ways to kind of knock out an astrocyte specifically or selectively. And he looked at it in the context of glial scarring with a stab wound. So he did wild type versus knockout astrocytes. And so the astrocytes are ablated at the wound site, and he wanted to see what would happen. And so what he found was that if you didn't have a glial scar, you found more trafficking of leukocytes, more white blood cells coming in. You had an impaired blood-brain barrier in the knockout. You had increased hippocampal neurogeneration in the knockout, which was glutamate dependent. So you had more neuronal death. But you saw increased sprouting in the knockout and more neurite outgrowth. So this last one's sort of a positive here. So we can just kind of go through this. The slides here show you the different sort of hippocampal survival. The axon sprouting regeneration is shown here, I think showing you in this example. So that's actually a good thing. If you want to reconnect, you want this axonal sprouting and neurite outgrowth. So the scar is important for a lot of things, like kind of limiting the damage and preventing neurogeneration, but it also prevents neurite outgrowth. So he's continually working on this complex problem of trying to understand glial scarring. So in this paper, this was 2016, they had a similar system, same lab, and he found that preventing astrocyte scar formation significantly reduced axon regrowth. RNA sequencing revealed that astrocytes and non-astrocyte cells in SCI, this is spinal cord injury lesions, express multiple axon growth supporting molecules. Our findings show, contrary to prevailing dogma, astrocytes, I can't see it because Garmer information aids rather preventing axon regeneration. So we kind of turned course here, about 20 years later almost, that now showing that the astrocyte scar is important for neurite outgrowth. And what becomes important from this is that it's really about understanding maybe the type of astrocytes that are involved, and sort of a good astrocyte versus a bad astrocyte, so to speak. And this is something that's gained a lot of attention. This is something that Ben Barris' lab was working on for a long time, this idea of different activation states. Shane Liddelow, who gave a talk, I think last year at OSU, maybe it was 2 years ago, another really great astrocyte biologist, talking about this, inflammatory astrocyte. He calls it an A1, this reactive astrocyte, and then this A2, which is a damage or ischemia-induced astrocyte. And so this idea of different populations of astrocytes that might be doing different things in the context of age and disease. And we'll definitely see this and some of our papers, and another probably pretty cool paper topic to talk about the different phases of astrocytes and their role in growth and repair. We'll also see this in the context of aging and disease, where astrocytes and different phases of astrocytes seem to support or modulate microglia, where we find that things like IL-10 and TGF-beta have this sort of cyclic response to reduce microglia responses. And we'll go through more of this in detail. This is, more of that reactive astrocyte idea with these different profiles, showing that, these reactive astrocytes can be toxic to neurons. So we definitely want to understand them and to do things to prevent their reactivity. Okay, so for the last, I guess we'll only go 8 minutes before we stop, and then we'll continue on Tuesday, right? So I wanted to get into the cell types that are really more immune cells, and these would be the microglia of macrophages, and they can present antigen, and we'll talk more about that later. But just to show you the spatial distribution, again, a term that we've used before, the brain parenchyma, this is where you're going to find the microglia. This is our representation of the blood-brain barrier. We see monocytes that come from the periphery can come into this perivascular space. And again, that's that space between the endothelial cells and the glial limitin. Now, what's really cool here, and we've actually had him come and talk to us at OSU and in class, this guy, Dorian McGavran at the NIH, has used 2-photon lasers imaging to see that this immune surveillance where these monocytes go into these perivascular spaces. And it's such a tight space that you actually see structural changes in the monocytes. They differentiate into this macrophage-like profile. That's what what this symbol is showing you. And then they get turned into like a worm or a tube-like cell. So the cells inside the perivascular space don't look round anymore. They look like a tube because it's the pressure, that tight space. So they become even a specialized cell. And these cells can go in and out of those perivascular spaces. And you think about, the immune system and immune surveillance, this is an important way that a monocyte can come in and sense what's happening within the brain. Remember that as waste is being removed from the brain, it's being passed over these perivascular spaces. Okay, I keep pressing the wrong button. Okay, so microglia, really, I think in terms of neuroimmunology, the most popular cell, and probably not any of the neuroimmunology papers that we'll talk about will exclude them. I mean, almost all of them have something to do with microglia. We'll see them in the blood-brain barrier. We know they're important for immune surveillance, structural support, also help support neurons and oligos. Microglia make about 15 to 20% of all glia and represent about 10% of the total CNS populations. In rodents, the ratio is about 2-to-1 gray matter compared to white matter. And in humans, it's the opposite. So you see a lot more white matter microglia than gray matter. And again, the white matter is where you're going to be seeing all the white matter tracks, right? All the myelinated axons. So it's interesting that it's this species difference that we see. As we talked about in the very first slide I showed you today, the lineages of these embryonic yolk sac myeloid cells. And they come into the brain, and you find them in the white matter and the gray matter. And in terms of detection, people use a lot of macrophage monocyte markers, things like CD11B, colony signaling factor receptor. This is fractalkine receptor. Another one that's popular in the brain is this IBA1 or IBA1, depending on what country you're from or how you like to say it. This is an intercellular calcium protein. And then these purinergic receptors. And then there's some newer things that people use now, SAL1 and TMEM119. They're supposed to be more microglia specific. Because one of the big things that with microglia you'll find is that a lot of the same microglia markers are also on macrophages. So if you have that question, if is it a macrophage or is it a microglia, then you can't tell by some of these normal stains or labels. So I mean, really, almost everything the microglia does is in the context of neuroimmunology. So its normal surveying role, it's actually actively moving its processes back and forth. I don't know if you can see me moving my hands, but it's those processes are sensing the local microenvironment. They're sensing for changes. We see that in development, that they're very important for synaptic pruning and monitoring synapses. And in fact, I think I'll post this on the chat, but I believe on Monday, we have Dory Schaefer coming in from, I don't know, is it BU or BC? She used to work for Beth Stevens, and her work was that seminal paper showing that microglia prune some of those synapses and eliminate synapses in eye development. And so she'll be giving a talk noon on Monday here. So I think that's right. I'll check. I think it's next week, but she'd be great to listen to. That's just an aside. But when the microglia become active, we often see these structural changes, which is one reason people use histology to look at them. You see that the processes become thicker and smaller and more kind of crunched in. And this is really the form that you're going to see them producing cytokines, complement, reactive mediators. Really everything that a macrophage produces, you'll see a microglia make. So these inflammatory cytokines, the big ones, again, the ones that you see over and over again are IL-1, IL-6, TNF-alpha, anti-inflammatories. So you also see them producing IL-10, TGF-beta, a whole slew of chemokines. Chemokines are like cytokines, but they're involved in recruiting other cell types. They have the whole complement of immune receptors, toll-like receptors, cytokine receptors. They can phagocytize things. They can present things on MHC class 2. They can scavenge things. They can proliferate, although it's much more limited than what you see at the astrocytes. We see them interact with astrocytes. And the other thing I think I want you to know, if you look at this slide, is that this reaction is reversible. So unlike some of the other long-lived tissue macrophages, microglia that get fully activated don't end up dying, all right? So in other tissues, you get these cells to die, oftentimes actually by glucocorticoid-induced apoptosis. Here, though, we see a lot of things that are tightly controlling microglia, like neurons and astrocytes, and pushes them back to more of a surveying state. So it's a reversible reaction. There's one more thing I wanted to ask you about microglia. Oh, yes. Why do you guys think that they change their structure so uniquely?
Is it because they're highly migratory, especially in activated states, so when they retract their processes, it's easier to migrate to a site of inflammation or damage?
Yeah, that's the prevailing thought. Yeah, is that as it kind of, the processes are much longer than I'm showing you with my little stick drawing. So the idea that they're kind of pulling them in for better movement. I don't know if you guys understand sort of the tightness of space within the brain, particularly within the gray matter. There's not a lot of space to move. It's pretty a tight network. So in order for a cell to kind of move and migrate, it has to change its shape pretty dramatically. So that's one of the ideas of why the microglia will spend all this energy to change their profile. And you see this. This is an inactive microglia, surveying microglia. It's expressing a lot of markers you'd find on macrophages, the CD11b, CD45, or something you'd find on a monocyte macrophage, lectins, some inflammatory receptors. And you see here, this is actually from a human brain. So you see this really big change in structure. The cell body is denser. These fibrous things are brought in tighter. You'll see some changes. So you'll see an inducible, especially in humans, CD11C induction, which looks more like a dendritic cell, MHC class 2, which is involved in antigen presentation. This is where you're going to see induction of a lot of different things like scavenger receptors. Some of you may have seen CD68 before, toll-like receptors, chemokines, complement, really can become more enhanced in an active profile. And remember that this population here is this is reversible. So it turns out there's a lot of diverse states of microglia, and this also applies to their cousins in the periphery, the macrophage. We have this, what's called the surveying or ramified because it has so many processes. Oftentimes we see this activated state, we see this deramification, meaning that they pull their processes in. The activated here is in red. And then we can also find microglia that are phagocytic, and they tend to look more round by histology. This is be ones that are kind of activated, picking up debris, and have a different profile than these cells here. And then in terms of what we also can find, and some of you may have heard of this before, is this kind of state that's intermediate, somewhere between the surveying and activated, not quite active and not quite surveying. And so oftentimes people call this primed or sensitized or reactive microglia. And you will definitely see that the more you read the literature, depending on the context, but certainly this other sort of state that you see. And it may just be an intermediate state that you catch the cell in between, going from a ramified to deramified or going back. There's this maybe should be the last slide that I talk about today, because it looks like we are right at the time that I'll stop lecturing. So we'll just do this one last slide, and then I will continue with this on Tuesday. So there's a lot of this idea of the different profiles of microglia. And so the astrocyte one that I mentioned, the A1 and A2, really comes from this idea. And this was all developed in macrophage monocyte tissue culture, and by giving them different agents that change their profile. And a lot of people absolutely hate this M1, M2 thing. Sometimes you write a grant and people will, pardon the phrase, will pee all over it. They won't like it. Other people like it. But the reason to teach it and the reason to bring it up is that you can't get away from it. You'll still see it. And the reason to teach it as well is that it does allow you to start to think about putting microglia in different categories, because to call it just activated or surveying or primed is too limited, right? And if we really want to understand disease and we want to cure disease and make therapies, we need to have a better understanding of what these profiles really mean, because sometimes these profiles might actually be very helpful in restoring and helping repair, as opposed to just causing neurodegeneration. And that's the classic problem, I think, with people getting into microglia biology and viewing them as a cell that just is a suicide bomber and is destroying everything around it, which is not a good way to think about what microglia are doing. So anyway, long story short is that you have this M1 profile, which is this activation. So the classic activation of these microglia with the pro-inflammatory cytokines and chemokines, that's called an M1. Then they have this M2 microglia, which is this alternate activation. So this M2 might have more of a tissue repair profile. Things associated with this were arginase, mannose, YM1. Then you have this M2B, which is the inflammatory and homeostatic. This is a microglia that's making both IL-1 and IL-10, and I can tell you can see that in a number of conditions. And then you have an M2C, which is a really anti-inflammatory or classical deactivation. And the idea is that this cell, once it becomes active, we know that it's tightly regulated and comes back to more of this anti-inflammatory. And we will talk a lot more about these profiles in some of my regulation lectures. Okay, so I'll stop sharing and we'll stop, let's see, I'll stop recording hopefully too. And I'll